Hi, I am Dr. Franziska Elmer, and in this video I present you how fast stony coral tissue loss disease spreads across Agaricia corals. Stony coral tissue loss disease is found on many Caribbean reefs and is a very infectious and often lethal disease that affects over 20 coral species, and infected corals have lesions that spread rapidly, leaving behind white coral skeleton. In this picture you can see the lesion edge and the coral skeleton. Agaricia are a very common coral genus on our study site, South Caicos, and on many other Caribbean reefs. However, because they are not common in Florida, where the disease originated, the spread rate of stony coral tissue loss disease is not yet known for Agaricia corals. The aims of this study was to determine the spread of the lesions on Agaricia species, compare it to other coral species, and examine the shape of the spread on the corals. Coral colonies with stony coral tissue loss disease were found, tagged, and filmed on the water. 3D models were made using structure for motion photogrammetry, and the area and circumference of the entire colony and lesion were measured in the model. The lesions on 11 Agaricia colonies were tracked in November 2019. They lost the tissue at a rate of 2.17 plus minus 2.45 square centimeters a day. This tissue loss rate did not correlate with colony or lesion size and did not differ among sites and depth. Lesions on 3 Diploria labyrinthiformis and on 5 Orbicella colonies were also tracked over time. Tissue loss rate was significantly higher in April than in November, and on Diploria labyrinthiformis and Orbicella colonies compared to Agaricia colonies. Furthermore, large colonies had significantly higher tissue loss rate than smaller colonies. Unfortunately, these independent factors correlated with each other. In April, both Orbicella and Diploria labyrinthiformis were surveyed, while in November most surveyed corals were Agaricia, with a few Orbicellas modeled. Corals were also larger in April than in November. The shape of spread was rather uniform on 2 surveyed colonies and variable on the other 17. Here you can see 4 colonies on which the first spreading edge is outlined as a dotted line, the second as a solid line, and on the top right colony, the third spreading edge is in white. On the right, the 2 colonies with the largest variation in spread are seen, and you can see that the edge is not advancing in some areas and advancing fast in others. Compared to other coral species surveyed in this and other studies, stony coral tissue loss disease spreads rather slowly on Agaricia. Agaricia is not often affected by coral diseases and may be slightly more resilient to stony coral tissue loss as well, despite getting infected. Despite our results being inconclusive on which factors affect the tissue loss rate, it is likely that the difference in tissue loss rate was mostly due to survey months. In April, 3 months after stony coral tissue loss disease was detected, 3 out of 5 surveyed colonies had very rapid tissue loss, much higher than averages documented in other studies. And 4 other surveyed colonies were completely dead when surveyed a second time. The tissue loss rate of Orbicella in November was a bit lower than reported in a previous study. It is therefore possible that the tissue loss rate in Agaricia reported here are also on the lower end of the spectrum. On most colonies, stony coral tissue loss did not spread evenly. We therefore recommend against using the nail method to measure spread, where a nail is placed at the disease edge and the distance to the new edge is measured afterwards. Surface area measurements, as done with structure for motion photogrammetry here, will be more precise and have become affordable in recent years, and are therefore our recommendation. I would like to thank all the staff and students from the School for Field Studies in South Caicos who helped with data collection, data analysis, and write-up for this study. This would have not been possible without you.